recharge from a little mini break. The goal today is just to answer questions about the respiratory system leading up to the exam that's on Thursday. Once again, the exam will be available at 10:30, 10 o'clock in the morning on Thursday, but if you prefer, you can take it during the lab period. You don't have to let me know. I just assume that there will be people who will take it in both times, and I'll make it available both. So, you know, this is the Moodle page, right? This is just everything that you've been working on, starting with key concepts, and then the PowerPoints, and the quizzes, and the answers, and the problem sets, and the answers. Questions? Yeah. So let me pull up the PowerPoint that triggers in my memory. Let's see. Go ahead. Did I already give you your mouse? Okay. That's right. So I can't remember exactly where this was. Okay. Let's try this one. Right, so this is what you're thinking about. This is the slide I was talking about when that question about ventilation, perfusion, matching comes up. The concept is that smooth muscle can control both the blood flow to a capillary bed by controlling the diameter of arterioles. You learned that in the circulatory system part of the course. And smooth muscle can control airway diameter to a specific cluster of respiratory alveoli. So, and really the main concept is that there are ways, the body has ways, the lung has ways to detect whether the perfusion of alveoli by blood is well coordinated, matched, with the ventilation of alveoli by the airway. And so, for example, and that's the main concept. You should know, that's the main concept. The idea is that if there is a part of the, let's just go through this, because it's in your textbook. If there is a region of the lung that is blocked by mucus or by asthma, then the blood in that region will not be getting, well, the airways in that region would not get ventilated, so that the blood that perfuses those parts of the lung would not get oxygenated, so blood oxygen would go down, and that would trigger constriction of blood vessels so that the blood could go somewhere else that was being ventilated. That's decreased blood flow to that region. That's maybe what's missing here. Not decreased blood flow everywhere, but decreased blood flow to that region, so that blood is diverted to the area that is not getting ventilated, to the areas that are. And so this is one way of matching blood flow to ventilation. Or if there was a region, let's just say there was a region where the blood flow was blocked by an embolism, or a clot, blood clot, some abnormality.
so that there was a part of the lung that was getting ventilated just fine, but blood wasn't going to it. Well, then there would be a detector there in the alveoli that would detect that PCO2 is in those parts of the lung that are getting well ventilated, but not getting perfused with blood, so they're not receiving the metabolic byproducts from the body. They're not getting CO2. So PCO2 would go down, and that would trigger constriction so that the air would get diverted, decreased airflow to those regions of the lungs, and air would then get gas would get diverted from the regions that are not perfused to the regions that are, so that the alveoli that are getting ventilated are also getting a little bit, more blood, matching the ventilation and the perfusion. So you don't need to be able to regenerate this figure. The main concept is that smooth muscle constriction of either pulmonary circulation or of airway can divert air or can divert blood or air to the areas where it serves the body's needs best. That's the main idea. I'm waiting for questions. Yes?
Can we go over the detailed steps for carbon dioxide, how it could enter the...
Okay, sure. Let's go over the story, the carbon dioxide story. So, let's see, it might even be in this. Yeah. Oops. Okay. This slide. Right? This slide. Okay, so let's walk through it in the kind of gory detail that I might expect from you if I asked you to tell me the story of CO2 removal on an exam. I would ask, if I asked a question about this, I would ask for, as much detail as you were capable of providing. So I might ask, where is CO2 produced? Yes, it's produced in cells, but where in cells is CO2 produced? Mitochondria. Okay, then mitochondria produce CO2 as part of cellular respiration. Okay, then this arrow. Let's get from CO2 produced to dissolved CO2 in the interstitial fluid. This arrow has quite a few steps that you could unpack. Tell me a few. I think I heard the word. Diffusion in cytoplasm. Diffusion through the membrane. Just Corey, give me everything. Diffusion through the interstitial fluid. Okay? And so CO2 is in the interstitial fluid. It's dissolved. It's a solute. Then this arrow. What would this arrow be? Let's unpack that arrow in all its fabulous detail. Diffusion into the endothelial cell. Diffusion, if you wanted to, you could say diffusion through a very thin endothelial cell cytoplasm. Diffusion through two endothelial cell membranes. That would all be part of this arrow right here. If you just said diffusion through endothelial cell, and you made it clear that you knew that cells were surrounded by a membrane from some other part of the story, I would probably say, okay, that's okay. Okay, so we're now CO2 has diffused into the plasma, the blood plasma,
inside a capillary. Some of it is dissolved. Dissolved solute. Some of it diffuses through the red blood cell membrane into the cytoplasm of the red blood cell, where it is dissolved. Some of it diffuses to, some of this dissolved CO2 binds with hemoglobin, so that's what that plus sign, binds with hemoglobin to form, carbamino hemoglobin. Carbon dioxide can bind to hemoglobin. So some of it, so far, some of the CO2 is dissolved in the cytoplasm of the red blood cells. Some of the CO2 is dissolved in the blood plasma. And some of the CO2 has bound to hemoglobin. Some of the CO2 that was dissolved in the cytoplasm of the red blood cell reacts with water to form carbonic acid, and that reaction is accelerated a millionfold by the enzyme that is present in the red blood cell membrane, carbonic anhydrase. That's what's going on here.
Can you say that stuff again?
Yeah, I'll say that step again. Some of the carbon dioxide that is dissolved in the red blood cell plasma, red blood cell cytoplasm, reacts with water to form carbonic acid, and this reaction is catalyzed by carbonic anhydrase. I don't expect you to know the millionfold, that's a detail. But I do expect you to remember that carbonic anhydrase is an enzyme expressed inside red blood cells that catalyzes that chemical reaction. And now carbonic acid does what all acids do. It dissociates into a base and an acid. So now some of the CO2 has been converted into bicarbonate ion. That bicarbonate ion is exchanged, is a substrate for the chloride bicarbonate exchanger. There's plenty of chloride on both sides of this membrane, so bicarbonate can get out of the red blood cell and be a solute in the plasma because of the chloride bicarbonate exchanger. If the chloride bicarbonate exchanger didn't move this bicarbonate would be trapped here because it's an ion. It doesn't just, this arrow might suggest that it could diffuse, but that doesn't happen. There is no diffusion of bicarbonate through the red blood cell membrane. There is transport by a chloride bicarbonate exchanger. And so now some of the CO2 was converted into bicarbonate and is now being carried in the plasma as bicarbonate ion.
Is that active or active?
This transporter, technically, it is a secondary active transporter because it is an antiporter. But in actuality, both of these movements can be passive. So I won't try to trick you on tell me whether that's passive or active, because it's tricky. But the point is it's a transporter. It's a protein that moves bicarbonate down its electrochemical gradient and moves chloride down its electrochemical gradient. And it's not diffusion as the textbook the arrows might have led you to believe. Right? So This arrow through this membrane is diffusion, because we're talking about CO2. This arrow through this membrane is diffusion, because we're talking about CO2. This arrow is not diffusion, because we're not talking about CO2, we're talking about bicarbonate ion. Okay, so I would hope that your answer would include that level of detail. And then you would, okay, so let's continue the story. Then there's blood flow from the tissue to the lungs. And now you would basically, you could say, okay, everything I just told you runs in reverse. And you might tell me, okay, some of the bicarbonate can get transported. Again, this is not diffusion, gets transported into the red blood cell where it is converted by carbonic anhydrase back into CO2. That CO2 diffuses through the membrane, diffuses through the plasma, diffuses through the endothelium. I'm going through this fast, and you can, I mean, if you feel like your answer has already told me the information that I need to know, you can start to abbreviate. Anyway, CO2 dissolves through endothelium. Dissolves, there's a cell here. What's the name of this cell? Type 1 alveolar cell into CO2. I mean, now we're talking about CO2 gas. But I haven't told the whole story, right? Bicarbonate transport, bicarbonate conversion into CO2, CO2 diffusion, da-da-da-da-da, CO2. But let's also remember there was some CO2 that was just dissolved in the plasma, and it gets there too. There was some CO2 that was dissolved in the red blood cell, but it gets there by diffusion, just like the CO2 that we brought in this way. Some of the CO2 had to unbind from hemoglobin. So I mean, if you can tell a complete story over here, it'll be easy for you to tell the complete story over here because it's just running everything in reverse. So I started to go fast again, which I regret. So are there questions about where I started to go fast?
Does that current dissolve CO2 into CO2 in the alveolus? It goes from the plasma to the
Endothelium,
and then
type 1 cell, into the air, the gas, inside the alveoli. And in principle, you should be able to do the same thing for oxygen, except that it's really simple. Right? So I could, all the details I was telling you about. Diffusion, type 1 cell. So this is now in the lung. We're starting with the lung, not in the capillary. Not in the body. Diffusion, type 1 cell, endothelial cell. Diffusion through the erythrocyte surface membrane. Diffusion, binding to hemoglobin. Most of it, 99% of it. Again, I wouldn't ask for the 99%, but I might ask, just in this case, most of the oxygen is bound to hemoglobin. I would not expect that for this, because it's a sum situation. It turns out it's most, but it's only about 60%. So some of the carbon dioxide is dissolved in the plasma. Some of the carbon dioxide is bound to hemoglobin. That's actually the minority. Much of it is converted into bicarbonate and is transported as bicarbonate. But I want to be clear. I'm not going to ask you to know percentages or relative amounts of each of these ways of carrying CO2. But in the case of oxygen, I feel like I could expect you to use the word most, or the majority of the oxygen binds to hemoglobin. And then you run the story in reverse in the tissues. Oxygen unbinds, diffuses through membrane, diffuses through plasma, diffuses through endothelial cell, diffuses through interstitial fluid, diffuses into the cell. You know, I'm going through this fast because I think you know what I'm talking about. Diffuses through the cell membrane, diffuses through the mitochondrial membranes into the mitochondrial matrix, where it is the final electron acceptor for cellular aerobic respiration. So you could tell me a detailed story of oxygen too, and it has only slightly less information than the carbon dioxide. I mean, it's just slightly less complicated.
I have a question about, so in that cycle that we saw of the ventilation, like the kind of
The matching?
circulation diagram, this one.
Yeah, partial pressures of oxygen, CO2. Okay, let me pull that up.
Well, while I find that, let's see if you can, think of your question. Okay, go to a different PowerPoint. What's the date on that handout?
October 2nd.
October what?
Second. Does it have a title at the top of it? Yeah, the respiration, ventilation stages.
Oh, okay. Oh, there it is. All right.
So in that section where it's showing that alveoli,
Yes.
and so the partial pressure of oxygen is around 500, and then it goes down to 100 when it diffuses.
Right.
At some point I read that the partial pressure of carbon dioxide stays the same through the diffusion, but oxygen doesn't because the diffusion of oxygen is slower.
That's right.
But
So
I thought oxygen was a smaller molecule than carbon.
it is, but it just, so I can't get into the physical chemistry
Oh, yeah.
of why oxygen would diffuse more slowly through membrane. It's through membranes.
Oh.
So it's not diffusion through liquid, and it's not diffusion through gas, because their oxygen would diffuse faster, because it is smaller than CO2. It's diffusion through membranes, which is the rate limiting step for these diffusion steps. And that's why the A difference, just to use terminology that we used in lab, in this case would be 5, right? The A difference for oxygen would be 5 millimeters of mercury, because there's 105 millimeters of mercury of oxygen here, and there's 100 millimeters of partial pressure of oxygen here. That's a 5 millimeters of mercury partial pressure differential, and that differential is mostly due to the fact that oxygen diffuses more slowly, less readily through membranes.
So I think that membrane or other way would be incorrect to say that just the diffusion of oxygen is slower.
If you did, yeah, I mean, I'm not gonna, this is not physical chemistry. I'm not gonna get into the mechanism for why, but you're right, diffusion of oxygen in gas and diffusion of oxygen in liquid would be every bit as fast as the fusion of carbon dioxide in both of those spaces. It's through membrane where there's actually chemistry that makes a difference.
I have a question about the final question in the problem set.
Okay.
If I, yeah. If
Let me
for-
pull up my answer key. Okay, so this is the final question. Do we need to, should we look at it? I mean, I can pull that up off,
Yeah.
well, let's see if I can find it. What number of problem set is this?
Problem
set
set
three?
3, 6.
I'm gonna show you the answer to a problem set we haven't got yet. Oh, we're not seeing anything. This one?
Yes.
Okay.
I have specific questions on questions D and E,
D and E.
which I think I got wrong.
Okay. Describe in words what the kidneys are trying, quote unquote, to do when they respond to hyperventilation. So what the kidneys are trying to do is excrete bicarbonate. So that the, in this case, hyperventilation has a low pCO2, because in hyperventilation you hyperventilated off, you've blown off CO2. So this is a low pCO2 curve. And the kidneys respond by trying to excrete bicarbonate, so that the ratio of bicarbonate to pCO2 is closer to normal, which is what determines that the pH is closer to normal. So what the kidneys are trying to do is excrete bicarbonate.
Yeah, so the blood will have more bicarbonate? Or
Less bicarbonate.
Less?
Yeah. The concentration of bicarbonate in the blood will go down because the kidneys will excrete it, get rid of it.
Oh, excrete means get rid of.
Excrete means urinate away.
I
Your
thought-
bicarbonate's going down the toilet.
Oh, so now it's blood, like,
It's getting out of your blood and into the toilet. Yeah.
On one of our slides, the one that talks about reduction and buffering of acid, you said kidney controls excretion or retention of H+.
Yeah.
So how, like,
So the H+ story is complicated. The bicarbonate story is simple. Let's focus on the simple part until we get to the chemical.
okay.
I mean, we'll do this in some more detail when we get to the kidney, which is the
Yeah.
next section. So,
I feel like this is more just like from my mental like picture, but like the type 1 cells create the layer, like the.
in the alveoli, yeah, the
Yeah.
type 1 cell is responsible for the shape and the size of the
Right.
alveolus.
And then the surfactant that's excreted by the type 2 cell, like, forms around the, like, I'm just trying to like, how does it have a thin layer that can be diffused if we also have this monolayer
Well,
around it?
okay, let me pull up that image. Are there any questions about this?
Yes.
Okay,
you
let's,
have an answer actually?
yeah, another question about this. Oh, E, you said E, right? Put a star on the point that represents the status of question 4. Okay, let's go back to question 4. Question 4 is your professor, da-da-da-da-da. But he has normal arterial PCO2 and normal blood pH. He's no, I mean, even though he is exercising, his PCO2 is normal, and his pH is normal. So he's normal, even though he's exercising.
Never mind.
This is me, by the way. Right? So this is the position that is illustrated by question 4. He's has high ventilation because he is producing a lot of CO2, but the ventilation is matching his production of CO2 so that his PCO2 is normal and his pH is normal. No?
Yeah, I now understand why I got it wrong.
Okay.
Because I thought it's D. But
You thought it was what?
I thought E refers to D. But I guess I'm just too stupid for that.
Oh. Put a star on the point that represents the status of question 4 above. I'm not sure how I could say that any clearer.
Yeah, I thought that's part B is question 6. I don't know what I thought when I answered that.
Okay.
So,
So during the exam, if you have a question about a question, and you would like clarification of a question, please don't hesitate to come find me. Okay, well, we were going back to surfactant. Unless there are more questions about this. Let's see. Does anyone have the PowerPoint, like the handout from the PowerPoint where I actually discuss surfactants?
It's from 927.
Say again?
927.
Does it have a title at the top? I don't have the
Surfactant,
page.
muscles, pressures, and lung volumes.
Say again?
Surfactant, muscles, pressures, and lung volumes.
Okay. Here we go. So the idea, Zoe, is that the type 2 cells exocytose surfactant into a microscopically thin layer of fluid, water, that is in contact with the type 1 cell. So this is the type 1 cell. Let's forget the macrophage. This is the type 1 cell. Here are more type 1 cells. There's a thin layer of water because cells don't like to be in contact with air. So there's a thin layer of water that could have been exocytosed by some cell or whatever. And then a microscopically thin layer, I mean this is nanoscopic, right? This is a molecularly thin layer of surfactant that is in contact with that layer of water. Surfactant is a lipid-like molecule, and so it's amphipathic, using some of the language from Self-Is. So its polar head it's energetically favorable for its polar head to be in contact with that microscopically thin layer of water, and it's energetically favorable for its hydrophobic, non-polar tails to be in contact with gas, not in contact with water. So I don't know, this has to be an exaggeration in the size, because this membrane right here is made up of a bilayer of molecules that are this size. So you can see that the scale gets screwed up in the cartoon.
That might have been where I was
Yeah.
like confused.
Yes.
So how does surfactant keep the lungs from collapsing? Or like the alveoli? Is it just the like for?
It's the breaking of surface tension. It's the disruption. This is how.
Oh yeah, can you go over this again?
Sure. So I don't have my demo thing, and it wasn't working very well anyway, but it is physically true that when you have a bubble, which is kind of what alveoli are, when you have a bubble of certain size,
the pressure inside that bubble is inversely proportional to the radius of the bubble. That's a physical law. And so if a bubble is smaller than another bubble, and yet they're connected by air, by a pathway, this bubble will have a higher pressure than this bubble, because this bubble has a smaller radius than this bubble. Higher pressure, nothing to prohibit flow. All the smaller bubbles will collapse into one big bubble. And one big bubble has a lot less surface area than a million small bubbles. So in the absence of surfactant, alveoli collapse into one, into a small number of large alveoli. And so the surface area of those alveoli is insufficient for gas exchange that is necessary. So the way to get around the physics, the way biology gets around the physics, is biology introduces a molecule, surfactant, that decreases the numerator as the denominator decreases. So if a bubble is small, it's going to have more surfactant per square nanometer. And so the surface tension, so the surfactant breaks the surface tension of water. And a higher concentration of surfactant breaks the surface tension of water more. So as the if this bubble were tending to collapse because of the physics, its surface tension would go down also. And so the surface tension, so now the radius is going down, but the surface tension is going down, and so the pressure is not going down. And so there's no pressure differential. And so the small alveoli can coexist with the big alveoli because surfactant breaks the physical law by decreasing surface tension in proportion to the amount of surfactant. I'm trying to be precise about it. The main point is that surfactant reduces surface tension and prevents alveolar collapse.
Did we go over how it actually breaks the surface tension?
That's physical chemistry. No, we didn't get into that. But I don't know. How many of you have ever done the high school or the middle school, maybe even the elementary school experiment, where you take a beaker of water, high quality water, and you can actually float a small sliver of metal on it. Metal obviously doesn't float, but metal usually sinks. But if you carefully place a sliver of metal on the surface tension of water, it will be suspended it'll be suspended at the surface tension. The surface tension is holding that sliver of metal at the surface. But then you add a droplet of dishwashing detergent and the thing sinks like a stone, because the dishwashing detergent is a surfactant that breaks surface tension. You've seen water bugs that skitter along the surface of water. They're using little hairs on their feet to maximize contact with the surface tension. And if you took a water bug, and you put it in a beaker, it would sit at the surface, and if you added a little bit of detergent, it would probably start drowning. Detergent is a surfactant. Lipid is a surfactant. Did that answer your question?
Do you want us to be able to explain this physics of this equation?
No, I will not ask you to use this equation, but I could ask you to explain what good is surfactant. What does surfactant do? What's the function of surfactant? Why do we bother secreting it? What cells secrete it? Where is it secreted in the respiratory system? Those kind of details could be on the exam.
Do we need to know the ventilation equations, or are those going to be provided?
You need to know the ventilation equations and the alveolar ventilation equations. You do need to know those. You don't need to know the oxygen equation that we used in lab.
There's one on the, so I'm looking at the key concepts. There's an alveolar ventilation equation that relates alveolar ventilation with tidal volume, dead space, and frequency. That one you do need to know. There's an alveolar ventilation equation that relates alveolar ventilation with the production of CO2 and the partial pressure of CO2. But there's an alveolar oxygen equation that we used in lab that you don't need to know. That's the one that's, so I'm looking on page 2 of the key concepts. John, are you, and I have extra copies of the key concepts if anyone's lost here. But at the bottom of page 2 of the key concepts, there's a key respiration equation. There's an alveolar ventilation equation. The answer to that question is yes, you do need to know that one. There's an alveolar ventilation equation, again, but it's a different equation, and the answer is yes, you do need to know that one, but you don't need to memorize the number. You just need to know the proportionality. And then there's an alveolar oxygen equation, and the answer to your question to that one is no.
Can you talk about respiratory bronchioles?
Respiratory bronchioles,
And
as
what-
opposed to conducting bronchioles.
Yeah, well, I guess I just don't completely, I get alveoli and I get the conducting part, but we had it on that quiz. and-
Yeah. So, I mean, I'm not sure to what extent I will make a big point about this, but the point is there is a cluster of alveoli, the alveolar sac, or the cluster of alveoli, but there's this tube here is bronchiole, because it's not alveoli, and yet it has some bulges that are doing gas exchange. So the fact is, it's bronchiole that is doing gas exchange, so it's respiratory. Here's bronchiole, let's just go to here. Here's bronchiole that is just the same size as this one, but so here, this bronchiole is respiratory, and this bronchiole is conducting, because there are no alveoli here, there's no gas exchange. And so this would be the junction between conducting bronchiole and respiratory bronchiole.
I think that makes sense. So then, would there be mucus produced in the bronchiole, but then surfactant in those alveoli that are on it?
Yeah, you could, if you just, so is the question where is mucus produced?
Well, I was just looking at this quiz.
What did I take off?
No, I
Oh.
just remember being like, oh, I don't really know. And just kind of
Okay.
guessing, so, but this.
So this airway would produce mucus. And then, what's the name of the cells that produce mucus? Goblet cells, and then there are cells that move that mucus, but they want to move it that way, not that way. Those are the ciliated cells. And so these cells in here would produce mucus. These cells here probably won't produce mucus because that would be a mess for these respiratory alveoli. So you know, I'm not going to get to that level of histology, but the point is these cells don't produce mucus. These cells probably don't produce mucus, and by the time we get up here, they do.
Okay.
So the respiratory bronchiole do not excrete the mucus, but still they still have the ciliated cell, right?
Probably. Remember, when cells develop, you know, there's gray zones, and I'm not gonna ask you to be careful about the borderlines. But knowing that conducting airway, whether we're talking about conducting bronchioles, bronchi, you know, the bigger ones, the trachea, all of that conducting airway produces mucus and moves mucus. Respiratory airway doesn't. That's a key point. Any other questions?
So I'm looking at key concept right now, and
Looking at what?
the
Oh, the key concepts, yeah.
alter like hemoglobin affinity for oxygen.
Right.
So we know like more acidity, more CO2 and higher temperature would decrease affinity, but it was key.
I didn't talk about it. That's from previous, it's diphosphoglycerate. It's a metabolic, it's actually a product of glycolysis. But you don't have to. Yes.
I'm also looking at key concept on page 3, or
Yes.
at the first section, oxygen transported blood.
Yes.
Factors that alter the hemoglobin affinity for
Right.
oxygen. What is
DPG, it's diphosphoglycerate, and that's a product of metabolism, of glycolysis. And I would not hold you responsible for that, because I didn't talk about it this year. So I apologize for this. That's a carryover from a previous year where I went into a little bit more detail about glycolysis. In a previous year, I wanted to review cellular respiration. And it wasn't, I didn't do it this year.
Also, at like normal temperature, like not elevated temperature,
Ah.
would the bicarbonate, would CO2 have higher affinity for hemoglobin compared to the oxygen?
So you're asking, would high temperature increase the affinity of CO2 for hemoglobin? Or increase the affinity of hemoglobin for CO2? It decreases the affinity of hemoglobin for oxygen, but I don't think it has an effect if it has an effect on the affinity of hemoglobin for CO2, it's a small effect, because I don't know what it is.
But in normal temperature, will oxygen or carbon dioxide have higher affinity for?
Oxygen has a higher affinity for hemoglobin than does CO2 under all conditions. But CO2 can bind to hemoglobin, and in fact, the binding of CO2 to hemoglobin decreases the affinity of hemoglobin for oxygen. Well, everything would be reversed in low temperature. So low temperature would increase the affinity of hemoglobin for oxygen. So a lung that was getting highly ventilated would have a low temperature, right? Because room temperature is usually lower than body temperature. I mean, if we're lucky, we're not living in a 98.6 degree air. So that would help hemoglobin bind oxygen in the lung, because the lung is relatively cool. And if we're exercising, muscles would make a lot of heat, and that would make hot muscle, which would help the hemoglobin unbind its oxygen in the tissue that needs it, the exercising muscle. It's a small effect, but these things add up, and it's an interesting phenomenon that hemoglobin has evolved in such a way that acid, CO2, and high temperature help it let go of the oxygen, which is bound with high affinity. Yeah.
can you go over the control of respiration? And what especially the medulla?
Ah. So the point here, this was the last class, this was just before break, so I understand that this gets fuzzy. The point here is that we have chemoreceptors, chemo cells in our, that line the capillaries in the carotid artery and in the aorta. So the carotid bodies and the aortic bodies have cells that detect oxygen and acid. Yeah, so they also detect carbon dioxide. So that would be, it would be appropriate to annotate that the peripheral chemoreceptors detect oxygen, pH, and PCO2. There are other chemoreceptors in the hypothalamus, so that should be on this album. I'll annotate this for next year. But the central chemoreceptors are in the hypothalamus. And that is a detail that I do think I expect you to know, that the central chemoreceptors in the hypothalamus, central just means it's part of the central nervous system. And so they're in the hypothalamus, and they detect PCO2 and pH only. They don't detect oxygen. The medulla oblongata is the part of the brain that controls the muscles that drive respiration. That's the main thing. And I don't, if this was just before break, I would not test you on that. The phrenic nerve is the nerve that connects the medulla oblongata to the diaphragm, and that's neuroanatomy that I would not test you on.
Why would you release proton as one of the factors that over the affinity of hemoglobin to oxygen?
Why would I?
Why would you release our proton?
Not sure I understand. I mean, because it's true. Acid also, so there are things that acid
Oh, okay.
acid helps hemoglobin let go, dissociate its bound oxygen. So acid, carbon dioxide, and high temperature. Ordinarily, I would say I'm happy to answer questions between now and Thursday. I'm teaching and I am happy to answer questions between now and Thursday, but I am teaching a self-phys lab on Tuesday afternoon and Wednesday afternoon, so my time gets limited. So if you want to meet with me before Thursday, it's going to have to be, well, this is done. It's today and tomorrow I'm doing self-phys lab, and meeting means in the morning. If you have a question that I can answer by email, I can do it in the evening. I'm just telling you that this afternoon and tomorrow afternoon I'm booked. Okay, and like I said, I'm happy to try to answer questions as best I can. I'll try to do it by email if possible. All right. Have fun studying respiratory physiology and anatomy.